Okay, folks, if we can filter back in, take seats. Again, I continue to be amazed at the amount of decorum and obedience of this group. It's exceptional. So welcome back. As I said earlier, we're going to have the two remaining speakers in this session and then a panel discussion. Our first speaker after the break is Kári Stefánsson, who is the CEO and founder of deCODE Genetics. Kári, thanks for coming.
All right. Basically, what we're doing, the kind of genetics that we have been talking about today, is two data sets on diversity in sequence and data sets on diversity in phenotype. And then we are looking for non-chance association between data points in the two data sets. And I work in Iceland, where we have been sort of working with a situation like Mark Daly was describing in Finland for the past 20 years. And we have basically genotyped half of the Icelandic population with one of the Illumina chips. We have sequenced the whole genomes of about 20,000 Icelanders to at least 30x. And I want to emphasize that claiming that we are sequencing the whole genome is a little bit bold. We have have gathered a very large amount of data out of the whole genome. We have the genealogy of the entire Icelandic nation on our computer database going centuries back in time. And you could look at that sort of as a phasing instrument. We have imputed the variance down to a frequency of about 0.01% into 390,000 Icelanders, dead and alive. And we have a list of at least 16,000 Icelanders who are homozygous for a loss of function mutation in at least one out of 1,800 genes. And we have set up a phenotyping pipeline, and we have started to call these people in, trying to figure out what indeed is the phenotype that goes with these knockouts. I just put together this almost meaningless list on the airplane coming here, but we have an enormous amount of data on the Icelandic population. We have basically the health history of almost everyone in the country that we can cross-match with whatever data we have on the variant in the diversity in the sequence. And I think it is very important to recognize that we get out to these. We and others have been pulling out to these basically two types of variants. We have been pulling out common variants, which are the variants that that either came out of Africa or have been under positive selection. And they're the ones that place us on a normal distribution curve of physiologic function. And if you're at one end of the curve, you're at the risk of a disease, and if you're at the other end of it, you are relatively protected. It is important to keep in mind that these common variants have usually subtle effect on the risk of a disease, but you can have a confluence of them, so you can have a very high genetic risk, even though it accounts for very little heritability. And then you have the rare variants that disrupt the same physiological function. And we and others are currently very much focused on the rare variants because they very often give us specific genes that can then be, the gene proteins that can then be used as drug target. We at deCODE have made a little bit of a contribution to the discovery of common variants that affect all kinds of common complex traits. We have also started to pull out significant number of rare variants with a large effect on all kinds of diseases. And I think it is important to keep in mind that once you have gathered a very large amount of data like we have done, both on diversity in sequence and diversity in phenotype, you can begin to do things in real time. So if you have a question that comes up in a drug discovery process, when you're evaluating the target, when you're evaluating the potential effects of manipulating the target, it is extremely important to be able to answer questions in real time. And that's one of the things that we have been doing for Amgen of late. For example, they, like so many others, have a program with an antibody against PCSK9, right? And And the big question, there are all kinds of questions that have come up, both in the discovery and the development process. What are the potential implications of turning down, cholesterol dramatically? And we have in Iceland the arginine 46 leucine variant that has significant impact on, particularly on LDL cholesterol, has significant effect on the risk of coronary artery disease. And one of the questions that we were asked, can you see any impact of variants in the PCSK9 gene on the risk of dementia? One of the things that came up was that people were really concerned that manipulating the target would cause dementia. So we took this variant and we looked at it in the context of Alzheimer's disease, other dementias, and we saw no effect. And as I will show you later, we indeed are powered to pick up variants in the genome that affect the risk of Alzheimer's disease and other dementia. One of the concerns was also type 2 diabetes, and indeed we saw no effect on type 2 diabetes, and we saw a little bit of a trend on an effect on lifespan, which was comforting. I can give you another example of in our work on blood lipids as quantitative trait, in our work on coronary artery disease, etc., we pulled out another variant, pulled out another gene that gives a fairly good target. And as if you contrast it with the PCSK9, the variants in the PCSK9 have stronger effect on the level of cholesterol. It amounts to a half a standard deviation, only 0.2 standard deviations, the effect of this variant in this new gene. Actually, it looks like it confers a little bit stronger protection against CAD than the PCSK9 variant. And actually, it has a significant effect on extending lifespan. It's a greater effect than from the PCSK9 variant. So one of the big questions that comes up when people are looking for drug targets in the prevention of coronary artery disease and myocardial infarction is this relationship between effect on LDL cholesterol on one hand and protection against the CAD on the other hand. So as you can see here, there are variants that like the PCSK9 that falls on the straight line, and then there are variants that have more effect on LDL, genes that have more effect on cholesterol, others that have more effect in protection against CAD. So the big question is this deviation from this line, is it due to the fact that some of these variants are working through mechanisms other than LDL cholesterol, which is basically the only thing that I think is left when it comes to drug development, drug discovery and development in coronary artery disease. To Alzheimer's disease, which is I think gives us another example of the use another example of the use of genetics in the drug discovery and development process. And it was a few years back when the Obama administration announced that it was going to spend a significant amount of money to sequence genomes from patients with Alzheimer's disease to look for protective variants. And it happened to be the very same day that that announcement came out that we found a variant in the APP gene that confers almost a complete protection against Alzheimer's disease. And it's a variant that makes the APP a worse substrate for base 1. And it was important for several reasons, because the pharmaceutical industry had been working for many years on developing base inhibitors, and it wasn't difficult to develop an inhibitor of base, but the ones that were developed caused blindness in animal models. They had an affinity for cathepsin D, if I remember correctly. So the concern was, are the side effects, because this is going to be a medication that's going to be taken for a long time, is the side effect unacceptable, particularly since we have no proof of concept that if we inhibit base, that is going to decrease prevent against Alzheimer's disease or be a treatment for Alzheimer's disease. So indeed, we found this mutation that decreases the cleavage by base 1, and it protects against Alzheimer's disease, which is an interesting proof of concept. And I think that that discovery contributed to re-energizing the base 1 programs in the industry. What makes this even more interesting for me as an old neurologist is that we took this variant and we looked at it in the context of the nursing home population in Iceland, in individuals who had not been diagnosed with Alzheimer's disease, but everyone between the ages of 80 and 100 has a slow but steady decline in cognition. And we were able to show that this mutation slows down that normal cognitive decline of the elderly, which basically means that if you guys get your act together and you put together a good base 1 inhibitor, it's probably going to be put into the drinking water of old people like me. We also, in the process, we also discovered another variant that predisposes to Alzheimer's disease in the TREM2 gene, which is in many ways interesting because if indeed TREM2 is a useful target in Alzheimer's disease, it's a target that is outside of the blood-brain barrier, and mostly outside of the blood-brain barrier, is also in microglial cells. And if microglial cells if are replenished from the periphery, you may actually be able to approach the target outside of the blood-brain barrier, although the effects may be within it. The big question, once you have a potential medication against a chronic disease like Alzheimer's disease, the big question is, when do you begin to treat, right? Do you begin to treat people, once they are diagnosed, or do you treat people in anticipation of diagnosing? So one of the ways in which you can do this is to use genetics to select people into a clinical trial. You can, for example, use it on individuals who are homozygous for ApoE4. And this is just data that took us about 10 minutes to pile together from our population data on onset of Alzheimer's disease based on number of alleles of ApoE4. A second possibility would be to use the age at the beginning of cognitive decline as the point when you begin to treat. And here you see the cognitive decline in elderly individuals based on the ApoE4 alleles. So, I think I'm correct that there are several companies that are beginning trials on the basis of using ApoE as a marker, as a treating individuals who are homozygous for ApoE4. Then it is the big question: what are we missing? What are the things that would make us all more powerful geneticists, allow us to use genetics more powerfully in the industry? And one of the things that I think we are needing desperately, and the thing that is going to lead to a quantum leap, is the better annotation of intergenic sequences. And I think it is particularly important because most of us develop these common diseases, or are predisposed to these common diseases, on the basis of common variants, not on the basis of these sorts of Mendelian. Very few of us develop these Mendelian phenocopies of these diseases. So I think that we are going to get a better focus, sharper focus on the mechanism behind the disease as a public health problem through the common variants once we have the intergenic sequences annotated. So we know exactly what's going to happen. So I think that this attention on the rare variants is going to be transient. We are fairly soon going to begin to look at the common variants again. The second thing we need is a social acceptance of what I believe is an inevitable principle, that when we go and seek help in a hospital for a medical problem, the probability that we will get help is based on the assumption that those who came before us allowed information about themselves to be used to make discoveries that were then turned into methods to treat or diagnose. I think it is completely unacceptable in a time where we look at access to healthcare as human right, that we can go to a hospital and say, no, you cannot use information about me to make sure that those who come, the next generation can have access to equally good healthcare. We need longer reads. We are basically saying that we have whole genome sequences and we have this in tiny little bits of 120 bases. We need much, much longer reads. We need somatic sequencing. I'm absolutely convinced that somatic mutations, just on the basis of what we know about de novo mutations, germline mutation, it is inevitable that we are full of somatic mutations that are going to have effect if they happen to be in cells that undergo clonal expansion. And the last thing we need is that we need to understand the brain better to be able to understand diseases in all other organs. And I'll just finish with an anecdote in support of that view. We, a few years back, did a study of the genetics of smoking. This is just my beautiful language, which says genetics of smoking and diseases related to smoking. And we found a variant on chromosome 15 that is such that if you have it and you smoke more. If you have it and you smoke, you have more difficulties quitting, etc. Then we took this variant and we looked at it in the context of lung cancer, and it confers an odds ratio of 1.3 of lung cancer. Lung cancer in Iceland is a pure environmental disease. 97.5% of those who smoke, those who develop lung cancer have smoked for decades. 97.5%. So it's a pure environmental disease. Without this environment, you will not develop the disease, but you inherit a compulsion to seek the environment. All right? So there is no line of distinction between nature and nurture, and I'm absolutely convinced that the environmental component of almost all of these common diseases is to a certain extent going to be explained by understanding how the brain is directing us in this direction or that. Now we have found a variant that quadruples the risk of oat-cell cancer, all right, that is independent, oat-cell cancer of the lung, independent of the smoking. And then we have also these variants that affect behavior that have an impact on smoking. And however, when you think about the potential that lies in gathering all of this data and using the genetics in a productive manner, I think this picture here shows probably more clearly than anything you have ever seen how incredible potential lies in this. This is a picture of old Walt Whitman on the left-hand side, and on the right-hand side there is a bunch of leaves of grass. And the difference between Walt Whitman and leaf of grass lies in the differences in sequences of A's, G's, and T's. So imagine yourself what we can do once we have sequenced entire nations. So thank you.